The female reproductive system includes the ovary. During this lecture, I would like you to understand that, first of all, there are different organs that make up the female reproductive system. And I'm only going to describe the ovary in this lecture. In other lectures, I'm going to talk about the other organs. So during this lecture, I want you to understand what goes on in the ovary during the ovarian cycle. That during the follicular phase of the cycle, follicles are recruited and go through various stages of growth until they get to the ovulatory phase, where only a selected one or two, perhaps, ovulate. So please understand all the changes, the structural changes going on leading to the time of ovulation. And then after ovulation, the ruptured follicle forms the corpus luteum. Understand the structure of the corpus luteum, how it forms, and then what it secretes during the luteal phase of the ovarian cycle. Also, as well as understanding all the structural changes going on through both the follicular, the ovulatory, and the luteal phase, make sure you're aware of the hormonal changes as well. The ovary is very important because it performs two functions. It produces the mature gamete, the ovum, and it also participates in steroidogenesis, the production of estrogens and progestins. And they have very important functions in different organs of the female body. So it's important to understand that the hormones produced regulate various cells, tissues, and organs throughout the body. And we will mention these as we go through this lecture, and also the effects of these hormones on other organ systems in other lectures. I'm going to now briefly summarise the major organs of the female reproductive system. In this diagram, just orientate yourself a little. It's a little bit too difficult when you first look at this diagram and try and understand all the structures shown without first of all orientating yourself. Try and identify the bladder in this diagram. On the right-hand side, you can see the vagina labelled. And that leads to the cervix, the uterus, and then you have continuity between the lumen of the vagina and the cervix into the uterus, shown in dark red, and then continuity between the uterus and also the uterine tube. A long tube that goes out towards the ovary. The ovary is shown here in blue with various structures I'm going to talk about labelled. The bladder is anterior to the vagina, and so the left-hand side is the anterior part of the body and the right-hand side is the posterior part of the body. Right next to the vagina, and not illustrated here, would be the rectum. So in this lecture, I'm going to concentrate mainly on the ovary, but also summarise the uterine tube. In another lecture, I'm going to talk about the uterus, the cervix and vagina, and also the breast or mammary gland, and the external genitalia. Moving on to the ovary. Remember, it's the organ in the female that produces the gamete, the ovum. It's also the organ that produces hormones, steroidogenesis. And the main hormones produced will be estrogen and progesterone. Here is a section through an ovary. It's a histological section taken through the ovary of an adult. And on the outside surface is a cortex. The surface component of the ovary is surrounding a medulla, where as you look here in this section, you can see lots of little profiles of tubes containing very bright red components. They're blood vessels, and they enter into the ovary through the hilum, into the medulla, and those blood vessels will then form capillary beds which go out towards the cortex, towards the surface of the ovary. And if you look at the cortex, you can see little round structures, some tiny, some bigger. Some have clear lumens. These represent follicles, ovarian follicles, at various stages of follicular development, which I'm going to describe in this lecture. Again, on the left-hand side is our image or our section of the ovary, showing you the cortex and the medulla. For the remainder of this lecture, I'm going to concentrate only on the cortex, because that's where all the follicular growth occurs throughout the ovarian cycle. On the right-hand side is a high magnification section taken of the epithelial surface of the ovary. And it's named the germinal epithelium. It's a mesothelium consisting of cuboidal cells, wrongly called germinal epithelium because early histologists thought this epithelium gave rise to the cells that formed the follicles in the cortex. And underneath that germinal epithelium is a dense connective tissue layer separating the epithelial surface from the underlying cortex where all the follicular genesis is going to occur. And that dense connective tissue layer or coating is called the tunica albuginea. Here is a diagram and also a section through the cortex of the ovary. I want to briefly explain this diagram and the section, and then I'm going to move on and explain the various stages of follicular development. The central figure, labelled A here, summarises what occurs in the ovary throughout the ovarian cycle. It shows a series of very round circular structures, follicles, undergoing various stages of development during the ovarian cycle. The ovarian cycle is divided into 3 phases. The majority of the phases you see in this diagram, A, is follicular phase, where follicles undergo growth. They get bigger. And those growth changes you see on the right component of the diagram labelled B. These follicles go through a growth phase where they do a number of different things structurally. And I'll show you these sorts of structures as we go through this lecture. But there's a couple of main points to remember as we go through. And I think they're illustrated fairly well on the right-hand side of the diagram, figure B. Follicles go through stages where they develop an epithelial layer around the oocyte. That epithelial layer is called the follicular layer initially, and then as the follicle goes through a phase of growth, that layer becomes very multi-layered or stratified, and they're called then the granulosa layer, which I'll talk to you about later on. So that's one event that happens structurally as these follicles develop during the follicular phase of the cycle. The oocyte also increases its size. It's not shown that clearly here, but that's another event that we need to recall as we go through this lecture. On the outside of this growing follicle are 2 layers that form. They're called thecal layers. An outside layer called the theca externa and an internal layer called the theca interna. The externa is merely a connective tissue capsule around the follicle, carrying blood vessels into the theca interna. The theca interna, we'll see later, is a very vascular layer. It's also very important because it produces precursors for the granulosa cells of the follicle to produce estrogens. And the third change you see when you look at follicular growth is about day 6 or day 7 of the follicular phase of the cycle. Suddenly a number of these follicles are stimulated to grow larger. They're stimulated to proceed towards the process of ovulation. And what happens then is they develop an antrum or a space shown at the very base of the diagram on the right-hand side. It begins in a secondary follicle, and that antrum gets bigger and bigger. It accumulates with fluid. and the cells are pressed out on the outside, the granulosa cells. And that follicle is now a very large follicle capable of ovulation. But as we'll find out, only one follicle normally ever ovulates. So this is just a summary of follicular growth. One thing to bear in mind though, is that the majority of follicles that undergo this growth development never get to ovulate. And at various stages of follicular growth, at any stage, follicles can often just degenerate. In fact, most of the time they do degenerate, a process we call atresia. Go back to the middle illustration diagram now, and those little follicles shown there in the diagram of the ovary, after ovulation, the ruptured follicle, the one that ovulates, is converted into a corpus luteum. And that corpus luteum is going to produce progesterone. The follicles, as they grow, produce estrogen. So on the bottom left-hand side, you can see a histological section through the ovary. And I'm going to now go through the stages of follicular growth that I've just summarised, but actually show you pictures or images taken from histological sections of each of these stages. And there are three major stages I'm going to show you. Firstly, the primordial follicle, and they're the follicles that are there at birth. They're actually in the ovary prior to birth. And then I'm going to show you details of the growing follicles, the primary and the secondary follicles, and then finally, the mature or Graafian follicle. On the image of the slide, on the bottom left-hand side, if you look very carefully in that histological section, you can actually see details of all those sorts of follicles. The very large circular follicle you see in the bottom left-hand corner of this section is a large secondary or mature follicle. You can see the antral space. You can see the circular oocyte. And then if you look again at the section to the right of that large mature follicle, you can see another follicle. It's a very well-developed primary follicle. There's no antrum there, but you can make out a very clear oocyte with a very, very prominent nucleus and nucleolus. And then all the little tiny ones out in the cortex are going to be primordial follicles and very small primary follicles. Well, here's a histological section of a primordial follicle. It's an oocyte, and the oocyte is surrounded by follicular cells. These cells were originally stromal cells, recruited from the stroma in the cortex, and they wrap around the oocyte and they protect it. They're supporting cells. And then those stromal cells, when the oocyte starts development, develop into what we call follicle cells. Well, these oocytes, these primordial follicles, as I mentioned earlier, are present at birth. In fact, about the third month of fetal life, in the ovary, cells develop into oogonia, the germ cell, and these undergo massive mitotic activity. Until about the fifth month of fetal life, you could have 5 to 7 million of these oogonia in the fetal ovary. They're actually in the first meiotic division, on their way to becoming haploid cells containing half the number of chromosomes and half the amount of DNA through the process of meiosis. But meiosis is not completed in these oogonia until adult life. In fact, the final stage of meiosis is never completed unless the oocyte undergoes fertilisation, which is very, very rare. Now, of the 5 to 7 million of these oogonia that develop, by the time the child is born, the female child, there are about 400 to 500 of these remaining. In other words, there's a loss, an enormous loss of these during fetal development, until you're only left at birth with 20% of these oogonia and oocytes. And that's because those supporting cells I mentioned earlier around them in the fetal ovary die off. They withdraw their support, so the oogonia die. Now, of that 400 to 500,000 oocytes that are there at birth, you're only going to have in a normal reproductive lifespan of the female about 400 eventually ovulating. The reproductive life of the female begins at about an average age of 12.7 years. It's called the menarche. And then menopause, or the cessation of the reproductive life in the female, is around the age of between 47 and perhaps 55. Quite variable. But during that reproductive life, ovulation occurs once every 28 to 30 days as part of the ovarian cycle, part of the menstrual cycle that lasts, as I said, about 28 to 30 days. So there is this enormous attrition of these follicles, these oogonia, these oocytes. If we now look at primary growth of the follicle, it's illustrated on this slide here, showing you two sections through the ovary, two sections showing you a primary follicle at various stages of its development. First of all, when a follicle changes from being a primordial follicle to a growing primary follicle, the stromal cells adopt a cuboidal type epithelium that you see on the left-hand image and labelled. This is the time at which you now define that growing follicle as being a primary follicle. If it's only got one layer, sometimes it's referred to as being a unilaminar primary follicle. If it's got many layers, such as the one on the right-hand side, many layers is often termed the membrana granulosa, then we often call it a multilaminar primary follicle. Notice the size of the oocyte. Even though these are taken at different magnifications, during this phase of development, the oocyte can change from being about 30 microns in diameter up to about 50 to then 80 microns in diameter. So there is enormous growth also of the oocyte. We often term the oocyte as being a primary oocyte, not because it's part of a primary follicle, but because it's only in its first meiotic division stage. It has not yet even completed its first meiotic division, and it won't do so until ovulation, if the follicle ever gets to the stage of ovulation. Now, what happens during this stage is that when these primary follicles develop, they have this single layer, then they do that independent of the gonadotrophins from the pituitary. But then when they get to a certain stage, they're dependent on follicle-stimulating hormone coming out from the anterior pituitary gland. And that follicle-stimulating hormone causes these granulosa cells, that they're now called, to multiply, to massively divide. And as they divide, they give rise to the membrana granulosa you see there, multi-layers of these granulosa cells. And those cells also, under the influence of FSH, produce estrogens. Now, if you look very carefully at the capsule around the multilaminar follicle shown in this slide, you can just make out a sort of a capsule, a bit of tissue wrapping around the cell, the follicle. That's called the theca interna and the theca externa layers. Unfortunately, in a lot of histological sections, you don't see details of these separate layers. But as I mentioned at the very start, the theca externa is a connective tissue capsule. The theca interna is a capsule that is very vascular. And as development starts, as these follicles begin to secrete estrogens in response to FSH, they only do so because the theca interna cells, the internal ones that are around the follicle, they become steroidogenic as well. They develop little lipid droplets inside them, cholesterol droplets that are the precursor for making steroid hormones. And this theca interna layer, under the influence of luteinizing hormone, or LH, synthesize androgens. And those androgens pass across the basal lamina, separating the membrana granulosa from the thecal layers, and those androgens are then used by the granulosa cells to make estrogens because they don't have the enzymes to produce estrogens from the very precursor products. So there is this combination between the theca interna and the granulosa layers, one providing androgens and the other then using those to convert those androgens to estrogens. And as the follicle grows, then more estrogens produced. And therefore, during the follicular phase of the ovary cycle, estrogens will increase in their levels in the blood. Also, you can see in the image, in the picture on the right-hand side, a shell around the oocyte. It's the zona pellucida. It's a homogeneous stain, very brightly stained, covering around the oocyte. That zona pellucida is going to persist right through until after ovulation. It's a very important structure that protects the oocyte, but it also has some very, very important proteins within it. One of them is the attachment site or the attachment protein for sperm. When sperm finally get to try to fertilize the egg, they attach to these proteins on the surface. And then another component in the zona pellucida induces the acrosome reaction, the reaction whereby the sperm can secrete the enzymes required to then penetrate through the zona pellucida and then access the egg to fertilize that egg. It's a very prominent structure, as you see here. The follicular theca you see there, the theca folliculi it's called, it's reversed the name sometimes, that's a term that you often might see, and it refers to really the theca interna and externa layer that I described a few moments ago. Now we see a very large, mature follicle, a secondary follicle on the left-hand side. You can see the oocyte within it, and on the far right-hand side you can see a huge follicle. These are taken at different magnifications. And the mature follicle, or the Graafian follicle, as it's called, that it's about to ovulate, is very, very large. It can be up to 10 or even 20 microns in size. It's a huge structure. And because it's so huge, you don't often see sections through the oocyte as you do on the left-hand side. It's just a chance that when you section some of these large follicles that you actually see the egg as well, or the oocyte. Now on the left-hand side, the secondary or the large mature follicle, it's gone from a secondary perhaps to the large follicular stage. You can see it's accumulated fluid that's in that space. It's called the antrum space. And the antrum space actually pushes all the granulosa cells out into the periphery. Look at the oocyte. You can see the clear stained zona pellucida around it, the oocyte's bright pink stain in this section. The cluster of granulosa cells around the oocyte are a group of cells we term the corona radiata, the crown around the oocyte. This group of granulosa cells are going to persist even after ovulation. And the very inner layer of these corona radiata cells actually penetrate microvillus projections through the zona pellucida and contact the microvilli of the oocyte. So there is communication between the two. There's interactions and control mechanisms going on. And then notice that the oocyte and the corona radiata are attached to the other cells around the periphery of the follicle by another mass of cells. That's called the cumulus operis. So here's the antrum. There's the cumulus operis that I've referred to, and the corona radiata is shown around the oocyte. The Graafian follicle is shown here, as I pointed out, but I just want to mention a little change that's occurring just prior to ovulation. If we can imagine we could see the oocyte inside this Graafian follicle, I want to just describe a few changes. Firstly, during the follicular phase, as I said before, around the 6th day, waves of follicles are stimulated to go through this growth phase. But only one eventually is going to ovulate. The rest, as I said before, undergo degeneration or atresia. Something happens biochemically inside these follicles, and I just want to summarise a very important concept. Have a look at the membrana granulosa in this large secondary follicle on the left-hand side. Recall that those follicular cells, those granulosa cells, are secreting estrogens under the influence of FSH. They're secreting those estrogens because they get androgens from the theca interna under the influence of LH. Well, what happens just prior to ovulation? One of these follicles, or perhaps a couple of them, actually start to get receptors for LH in the granulosa cells. Those granulosa cells up until now have only had receptors for FSH, which controls their production of estrogens. Suddenly, about 12 hours before ovulation, or maybe even a little bit earlier, they acquire, or some acquire, the ability to have LH receptors on their cell membranes as well. And that's extremely important for two reasons. One is, one of these follicles is going to get more of these receptors than the others. And therefore, they're in a position to respond first, or best, to the LH surge that we see at the time of ovulation, that induces ovulation of one of these follicles. So the dominant follicle, if you like, the one that has the more numbers of LH receptors, can respond to that LH surge and therefore ovulate. But another thing also happens. Those LH receptors are also very important for the eventual destination of this Graafian follicle once it ovulates. Those LH receptors are going to be extremely important on these cells for those cells to convert to produce progesterone after ovulation, and therefore we enter the progestinal or the luteal phase of the ovarian cycle. And some of that progesterone gets produced by these cells just prior to ovulation. So those two very important concepts are very important to understand with regard to responding to the LH surge at ovulation and then finally being able to produce progesterone in the corpus luteum, which is what this follicle will develop into after ovulation. Again, you can see the thecal layers here. They're becoming more engorged with blood, which assists in also the ovulatory process, but also at the time of ovulation, these thecal layers, the theca interna anyway, is going to penetrate into the ruptured follicle. Up until now, follicular cells do not have a direct blood supply. They receive all their nutrients by diffusion from the theca interna into all the cells of the granulosa. But after ovulation, as we'll see, this theca interna layer, the theca interna, the very vascular layer you can actually see here quite prominently, will invade the developing corpus luteum and convert the corpus luteum into an endocrine organ. As I mentioned earlier, a lot of the follicles undergo atresia, degeneration. And you see certain signs of atretic follicles in the ovary. On the one on the left, you can see that the cells just don't look as pretty as they do in a normal, mature or developing follicle. There's a lot of cells that are lost and moving into the antral space. If you look on the right-hand side, on the very right bottom corner, you see these little sharp pink structures, shiny pink structures found in the ovary. These represent the zona pellucidas that have remained after degeneration of follicles of certain sizes. And again, on the right-hand side of the right-hand section, you can see a rather elongated pink structure. That's called the glassy membrane, because one thing that happens during atresia is that the basement membrane separating the thecal layers from the granulosa layers of the developing follicle hypertrophies. And during atresia, it gets thicker and thicker and rather amorphous in staining, and we refer to it as being a glassy membrane. These are all evidence of follicular atresia. Now here's a corpus luteum. On the left-hand side, this is an image, a section through a corpus luteum from a recently ruptured ovarian follicle, Graafian follicle. All the red stain material you see is blood, because when the follicle ruptures, there's some bleeding. And there's some growth of blood vessels from the theca interna into the corpus luteum mass. Finally, that corpus luteum will grow to be a certain size. And that antral space you see, which was previously part of the antral space of the follicle, will be converted into a very small fibrous core. And those blood vessels, as they grow in, as I said earlier, establish an endocrine tissue, an endocrine organ, whereby the granulosa cells have changed structurally and they've become vascularized. And they now have the capacity to produce progesterone under the influence of LH, which I described earlier on. And this progesterone now coming out of the corpus luteum suppresses the development of follicles in the ovary during this luteal phase of the cycle. My PhD was on the corpus luteum. I looked at the growth of the corpus luteum and I looked at how blood vessels grew into the corpus luteum, because one condition that prevents women from maintaining pregnancy beyond a few months is called luteal insufficiency. It's the inability of the corpus luteum to produce progesterone. So my research project for my PhD was actually looking at this structure. So I have a very soft affinity for it, an affection for the corpus luteum. On the right-hand side, you can see a high magnification picture of a part of the corpus luteum. The bluey, purply stained cells represent the previous granulosa cells of the follicle. We call them now granulosa lutein cells. The very paler stained cells you see represent cells from the theca interna that have invaded into the developing corpus luteum from the ruptured follicle. They're called the theca lutein cells. So you have theca lutein cells and granulosa lutein cells. And the relationship between these two cell types is similar to what the relationship was like when they were in the follicle. The granulosa lutein cells are now producing progesterone. They're also producing estrogens because the theca lutein cells again provide those granulosa luteal cells with the precursors for them to be able to secrete estrogen, just as it was in the follicle. Now the corpus luteum just means yellow body, because when you look at the corpus luteum in fresh ovary tissue, it just looks yellow. And that's because all those lutein cells have accumulated cholesterol stores, lipid droplets, that is important as being a precursor for producing progesterone and for producing estrogen. So that's why it's called the yellow body. When the corpus luteum has stopped functioning, it's because it has its support withdrawn from the pituitary. LH with support is withdrawn. And that happens about the 6th day after ovulation. Why does that happen? It happens normally because after ovulation, we need to think a little bit about what happened to the oocyte. At the moment of ovulation, or just prior to ovulation, the oocyte completes its first meiotic division and enters its second meiotic division, but is arrested. So at ovulation, the oocyte is quite large, about 150 microns in diameter. And you can see the second polar body alongside it, still enclosed in the space between the oocyte and also the zona pellucida. And that's a sign that ovulation has occurred. That oocyte, having entered the second meiotic division, is now called a secondary oocyte. And as I said before, it will only complete its second meiotic division after fertilization. That oocyte is collected by extensions from the uterine tube that I'll show you in a moment. It's almost like a erectile tissue. It wraps around the ovary at the time of ovulation so that the ovulated oocyte passes into the uterine tube and down the uterine tube for the chance of being fertilized. If that oocyte is fertilized, it spends another 3 or 4 days moving down the uterine tube until it finally implants in the uterine wall, in the endometrium, about 6 days after ovulation, during which time this corpus luteum is producing more and more progesterone in response to LH. But if implantation doesn't occur, that support is withdrawn. And it's withdrawn because as soon as the fertilized egg implants in the endometrium, then the endometrial cells secrete human chorionic gonadotrophin. And that immediately signals to the pituitary that there is a pregnancy, there is implantation. And that therefore maintains the secretion of LH, and therefore it maintains the lifespan and function of the corpus luteum for a number of weeks and produces increasing amounts of progesterone and estrogens that are so important for the maintenance of the pregnancy. But if pregnancy does not occur, implantation does not occur, then the support is withdrawn, as I said a moment ago a few times, and therefore the corpus luteum degenerates. And also because the support is withdrawn, follicular growth starts again. And that's the beginning of another part or another ovarian cycle. The corpus luteum then, after these granulosa cells start not producing progesterone, and as the theca lutein cells stop providing the precursors for producing estrogens, this corpus luteum starts to involute. These cells die off. And finally, you're left with a scar called the corpus albicans. And that finally again diminishes and is just a fibrous scar within the ovary. So let me now summarise what I've described in this lecture. I've named the various organs of the female reproductive system, and then I have concentrated on the ovary, and I've described all the stages of follicular growth, the follicular phase of the ovarian cycle. And we've seen how the follicle grows and then what hormones is secreted by that follicle. and what controls the secretion of these hormones from the follicle. After the ovulatory phase, the ruptured follicle develops the corpus luteum, and that corpus luteum is a very important structure secreting progesterone and estrogens. Know what hormones control the secretory role of this corpus luteum during the luteal phase of the ovarian cycle. And then understand why the corpus luteum on most occasions regresses, why the hormonal support for the corpus luteum is withdrawn. So thank you very much for listening to this lecture. I hope you now know a lot about the histological structure of the ovary and all the changes that occur within the ovary during the normal menstrual cycle and ovarian cycle. And I, again, thank you for listening.